Ching-Po. It's my great pleasure to be here to talk about basic idea for MRI and how can MRI be used to map brain function and brain structures. So let me have this slide to have the introduction. So basically, when we notice these beautiful ladies, actually, for me, I want to know what is she looking for, or what is think about, or even I want to know whether she loves me or not. So the basic idea is that, okay, so for us, we want to know how does she thinking, what's the thinking process, or what is the cognitive function of these beautiful ladies. So, and also we want to know what is the structure underlying these functions. So the basic idea for us is to know what is the brain function and brain structure. But the idea is that how can we detect that? The most important idea is to detect that non-invasively. I believe most of you, and including me, don't want to destroy this beautiful lady, right? Just after your detection. So the best idea for us is to have non-invasive detection. That's a very, very important issue. As Professor Peng just introduced that we must have such kinds of measurement, but non-invasive is the most important issues. So today I will talk about such kinds of measurement based on different kinds of modalities. Some modalities actually Professor Peng already introduced to you. So I'll more focus on the structure and functional, how can we detect that? Actually, nowadays, there are two kinds of methods we can detect, non-invasive detect brain structures. One is for X-ray. That's kinds of, actually the basic idea is, or source is X-ray. And of course, we can detect the signal from different directions. Then it gets the computer tomography. That's how we conventional talking about the CT. And it's kinds of radiations measurement. And the other one is MRI. MRI is actually a very powerful technique I will introduce more later. And so nowadays, the most convenient techniques for structural measures as CT scanner and MR scanner. So we can see actually this is the CT results. We all know that the source is X-ray, as kinds of radiation. So when there is X-ray just penetrate to the brain, then there are some signal attenuation due to the bone that is solid tissues or the soft tissue of the brain. So you can see that different kinds of contrast. However, for such kinds of X-ray measurement, actually you know that the contrast is poor. Right? In compared to MRI, actually the contrast is very, very poor. However, that is very convenient techniques. For us, when we have such kinds of whole brain measurement, that actually less than 20 seconds. So it's very convenient and also efficiency to have that in hospital and also for urgent detection, for example, for the patient who has ischemia. And for us, we know that for the patients who is underlying, who have stroke, actually time is money. Time is very, very important to the patient. So we must have some informations in very short periods. But for us, to have the cognitive measurement, or we want to know the brain functions, actually we still don't use such kinds of technique because actually there are some radiation. When you have a little bit scan, it's fine. 1 or 2 times per year is fine. But we can hardly have a long-term measurement, right? So another technique is MRI. MRI nowadays is the most, I can even say that it's the best structural measurement methods. You can see with MRI, we can have the details of the structure, for example, from the cerebral cortex to the white matter and different regions. And actually, such kinds of imagery is in 3D machine using the Siemens techniques in my lab. And we can have the brain structure in 5 minutes. It's in very high resolution. And based on that, we can actually measure a lot of information I will introduce to you later. And this is another technique I want to share with you that's based on the one kind of MR techniques we call diffusion MR. There's also another diffusion, another MRI technology that we can use to detect neural structures. That's both techniques I will introduce to you later. Further, let's talk about the function. How can we measure brain function? As Professor Peng just introduced that when we we want to know the input or output, the signal and how the brain processes that. The most important idea is that we want to have non-invasive detection. So how can we have a non-invasive detection? That means we must detect the signal outside of the brain, right? So when we measure the signal outside of the brain, then we must think about what kind of information that we can measure. So there are different kinds of information that we may measure outside. One is the electrical activity. That is more directly because that we know that when there is neural firing, then there is neural activity. So we know that for some reason, for example, when I'm talking to you, maybe there are some neural activity in my language area. So when we have some detections here, then you can know my neural activity. Then that's kinds of electrical activity. And also underlying the electrical activity, there are some metabolic activity that must happen to support such kinds of neural activity, and also hemodynamic activity, because we need to have more blood, or fresh blood to support the neural activity. So actually there are different kinds of activity happen there. So the basic idea for us is that no matter what kind of information that you detect, that means the area has brain function. So that's the basic idea for us. And of course, as I introduced to you, that we must have such kinds of non-invasive measurement. So nowadays there are a lot of techniques. I just summarize here. There is each kinds of techniques that we can use to measure the electrical signal. It's very simple and very easy use techniques. However, such kinds of techniques actually will be because the electrical signal transportation will be limited by the neural system. So even though we can detect the signal here in very high temporal resolution, but you can hardly know where is the neural signal firing. The spatial resolution in the brain is poor. Okay, so that's the kind of techniques. So that's the reason why there's another kind of MEG techniques. MEG techniques is actually just to measure the magnetic field. What is the idea? For example, when there is a neural, for example, here is a neural. If there is a neural activity here, we know that there is a magnetic field in here, right? So the basic idea is that for us, we want to know where is the signal source. So if we can detect a magnetic field, then we know that magnetic field transportation don't need to any medium. So we can exactly locate the source. But the problem is that such kinds of magnetic field is very, very small. Kinds of 10 to minus 12 to the Earth's magnetic field. So actually such kinds of magnetic field is very small. So we must have the shielding rule and also have a screen. Let's high temperature superconducting detection to have the magnetic field. So that's both kinds of methods that can detect neural activity in the brain. And the transcriminal magnetic stimulus is another kind of method. Actually using also such kinds of electrical measure, but it's on the contrary ways. That when we have the very short magnetic field and electrical signal here transport, then we know that once there are electrical change there may be a magnetic change. There is kinds of very basic physics phenomena. So we know that when there is a very short electrical change, there is kind of introduced magnetic field here. So when we see here is coil, then we can see there is a very short magnetic field change here. And for the neuron, it detects the very short magnetic field. Then it induces very fast electrical signal. So with these techniques, it can actually interrupt local neural activity. so we can also know what is the brain functions at this region. So here is the kind of very powerful method for electrical measurement. And for functional MRI and the near infrared also, they can kind of another techniques. But for both methods, actually they detect the blood function, the blood flow. For example, for a NIRS, actually detect the oxygen hemoglobin and oxygen hemoglobin because they actually have different kinds of wavelengths. So using optic detections, we can detect the both signal of the brain, and then we can reflect the brain function. So that's another technique. And MRI using similar source. So then actually I will introduce later. And PET is another method that actually can be used to measure the metabolic. But however, nowadays it's still be used to study brain function because it must be inject a radiation source. Like so actually there are some radiation. Of course, for normal measure, it's okay once per year. But nowadays, still people using that for major cognitive functions in healthy subjects. And here is the example for a EEG measure and MEG measurement. So you can see that with MEG measurement, we can locate the region by some mathematical method and to localize the source. But of course, in contrast to MRI, you can know that even though we can identify some regions, but the spatial resolution is poor to MR. However, for EEG and MEG, the temporal resolution is very high in compared to MR techniques. And here is the PET result. It had a very good benefit. We can detect a specific metabolic source. For example, you can detect glucose and so on. And here's functional MRI. And later, I will more focus on functional MRI because actually that is the techniques nowadays we use very easily and can detect a lot of information. We need MR. So here is the MRI. Actually, MRI show different kinds of contrast. For example, T1 and T2 actually is kinds of physical characteristic. So for us, the idea is that to differentiate the brain structure just based on their physical properties. So T1 is kinds of signal relaxation properties, and T2 is related to spin speed. That means the local magnetic field changes. And also diffusion is another technique I will introduce more later. And the susceptibility is based on the local inhomogeneous of the magnetics. And functional MRI I will also introduce later. And MR is the techniques that can detect local frequency change of based on the underlying chemical source. And this is MR angiography. Then we can see the vascular system. So actually, MR have different kinds of ways to detect different kinds of source. But due to the time limits, I will only introduce to you to T1 anatomy information and diffusion that can be used to detect neuron tracks and also functional MRI. So we need the lecture. And this is the idea, the example for anatomy used for clinical study. For example, when we have the anatomy information, and for tumor patients, we can see it can easily identify the tumor region in this region, or for the patient with ischemia. And MR is nowadays the most standard techniques to identify this ischemia. And even though we need only within 20 minutes, we can actually identify the region, as usual here. And here is an example for dementia patients. You can see the atrophy of hippocampus in these regions. And also that's how hypertension means that this technique can be used to detect the abnormality of hypertension tracts. And also there's a lesion in drug abuse that we can identify some regions. So for these techniques can provide very detailed structure changes. So that's also the reason why for these techniques nowadays being used for clinical study. But for us, when we considering for structural informations or what's the change, so for example, following aging or following the change of dementia, then we can see such kinds of technique also can provide some information. For example, this is a young population, and we can see the structure is here in compared to the patient with schizophrenia or dementia and so on, or even with some aging populations. We can see actually the brain changes because the brain entropy, but the entropy location or the value of the change actually have different kinds of meanings. So for us, the basic idea is that we can see the changes, but how can we detect that? So that's the idea. How can we detect? Yes, we can see the changes, but the problem is that once the patient come here, or even when we have a study, you can hardly have the image of the same people before 5 years or before 10 years. That means you have no baseline. So for us, when we want to have such kinds of measurement, we need a baseline to know what's the difference, right? So to have the baseline, the basic idea nowadays is to have group analysis. That means, for example, I want to detect dementia patients, then I must have a group of dementia patients. Then I must have a group of normal subjects. Because, for example, if I am a healthy subject, then my brain is actually different to yours, all of you, and I know most of you are healthy subjects, but our brain is actually different. So we can hardly use only one brain to be the template. Do you know what I mean? Because the change belongs to the gene difference, or linked to your education or thinking process and so on. So for us, the more easily way, or the more convinced way, is to have such kinds of group analysis. Means that we have the patient in a group, and then normal subject in a group. Then we have both into a standard template. Then we have the statistic analysis. So that's the basic idea for us to have such kinds of study, to know what's the difference between two groups, or to know what's the difference in dementia patients. So for here, you can see that we must have a group of subjects, and then we must have a lot of signal processing to register the brain into the same brain and into the template, and then identify the regions, different regions, for example, 5 meter, gray matter, and CSF, and then have the mask to identify the specific regions. Then we have all localized, then we can see the difference. So you can see for nowadays that for such kinds of brain study, not only just to acquire good images, but also need very good post-processing techniques. In this way, you can identify the very important informations within these two groups. So I want to emphasize that. So in my knowledge, that I noticed that such kinds of post-processing nowadays is very, very important, and a lot of different kinds of methods that just be developed to identify or to find out the most subtle change in the brain. For example, for anatomy, that we can identify the change of brain balance, or even for the stalkers, or for the cortical sickness and so on. And for diffusion, I will introduce to you later. That also can be used to detect the 5 beta tracks, and also can be used to have whole brain network analysis. And functional MRI is also another technique that can be used to detect brain function. We must have a very detailed or very solid statistic calculation to identify the regions. And here I want to also emphasize one more information is that different methods actually that's sensitive to different information. That's very important. So for example, someone told me, asked me, I want to detect gray matter balance. I will tell you just choose T1 anatomy, because it has very good contrast between gray matter and white matter. But if you ask me, I want to detect white matter tracks, I want to detect white matter change, I will tell you, you can use diffusion techniques because this technique is sensitive, highly sensitive to neuron change, especially in bimeter. But diffusion, whether diffusion can be used to measure gray matter, yes, but sensitivity is poor. Okay, so for us in engineer, I will more focus on such kinds of techniques and what's the benefit between two different methods, and then what is the best way to be used to answer the question. That's very important. Okay, then here is the example of the IQ intelligence. That's a paper published in 2006. We can see that in that paper, they just measure the IQ performance of different people, and they noticed that such kinds of IQ performance actually link with brain volume. And of course, they use such kinds of cortical measurement. And also in this paper, they also emphasize that for some people, they are very smart, they can, so genius. Actually, they may not so smart when they are young, but actually they just keep developing. So in this paper, they're talking about such kinds of issues, and the basic idea is using MR to detect the brain structures and have such kinds of measurement and comparison. So here's an example. And here's another example that they study the brain atrophy in elderly, and we can see following the age changes, the brain volume actually changes. And here is male and female. And here is another is total brain volume, and so on. And this is how many changes. So based on such kinds of idea, we can detect the changes of brain volume or even in patients. You can see, and here's another example that study in our group that we can see the genetic effect in gray matters and also their cognitive performance. Then we know that different kinds of the genetic performance actually link to brain structures and link to cognitive functions. So here's another kind of example, and this technique also used the techniques I just introduced to you is anatomy measurement and also have the volume calculation. And here is another example in Alzheimer's patients. You can see that in these patients, there are different atrophy in different brain regions. So based on such kinds of techniques and also the post-processing, we can identify the changes or even the progression. I mean, the following the onset of duration, and what's the volume changes. So that's the basic idea for us to use such kinds of techniques. And then I will introduce to you about the other techniques we call diffusion MRI. That is the only method nowadays can be used not to non-invasively detect neuron tracks in the brain. And here is an example that is internal capsule. And you can see that there's kinds of model from postmortem brain, and nowadays we can acquire the signal non-invasively in the living brain. So this is the very powerful techniques nowadays to detect the neural tracks. And the basic idea is that for these techniques, you can see, for example, there is a diffusion. We can see a dye when we have the dye drop into the cup. Then after a couple seconds, then we can see there's some diffusion. So following the time changes, then we can see this limitation of the environment. So that means the diffusion cannot happen outside. So you can see the diffusion limited in this environment and so on. But if there is no limitation of the environment, then we can see the diffusion just changes. That is kinds of short distance changes following the Einstein rule. So the basic idea for us is that we don't know what is the structure behind the blue mask, but if we can measure diffusion probability along different directions. For example, if we can measure the diffusion probability along different directions, then we can consider that, okay, maybe the structure behind the blue mask is an ellipsoid, right? So that's the basic idea for us. And MRI is a technique that can be used to detect diffusion changes. For example, here is a neuron, it's corpus callosum. And we can see in this here, the corpuscular sign is like just from left to right. So when we measure diffusion from left to right, measure that, we can see we measure the signal from left to right. Then we can see diffusion along the epsilon directions actually is changed a lot because it has higher diffusion coefficient. Then for diffusion line, actually is use signal attenuation to measure the diffusion. So we can see that the signal is stuck here. So and also in these regions, if we measure signal diffusion signal along this direction, we can see the diffusion in this direction actually limited by the myelin, by the structures. So actually the signal have less diffuse, so the signal change less. So you can see actually the signal have less attenuation, so the signal is bright. And here is through plane measure, so we can see through plane measurement also whole is bright. So based on such kinds of basic idea, we can see that if we can measure the diffusion signal along different directions, then we can map the neural tracks non-invasively. So that's the basic idea for us. And the most successful model was proposed by Peter Besser in 1994, and it have a very simple relationship to that, because at that time, just as I introduced to you, that we must measure a lot of directions to know the underlying structures. But for him, he just have a hypothesis that suppose that the diffusion along the neural tracks is the ellipsoid model. What's the mean? It means diffusion along the neural directions have higher diffusion probability, but perpendicular to the neural tracks have less diffusion probability. So that means he just have the ellipsoid model hypothesis. And from mathematics, he just show that, okay, in mathematics, we can just use a tensor to represent the ellipsoid model. So from that, we can see there is only 9 different number we have to measure. But you notice that diffusion along this direction is similar to, or we can see the same to this direction. That means it's a symmetry characters. So that means these two diffusion is the same, these two is the same, this two is the same. So for us, we want to differentiate the 9 different numbers. Actually, we just have to measure the 6 numbers. So for us, what we have to do is to measure different 6 diffusion directions and then 1 reference images. So that's the basic idea for DTI, and that's also the reason why it's so successful. Then we can have the ellipsoid model just from the tensor. Then we can see there is 3 directions that along the track directions, and also 2 perpendicular directions, and also 3 values. The values means the diffusion coefficient along different directions. That means different coefficient along different neural directions. So based on such kinds of idea, you can see that, for example, in the corpuscular side in the brain, now we can see in this region, the ellipsoid model is that kind of cigar. You can see because the signal diffusion along the tracks is higher in compared to the diffusion perpendicular to the other directions. So, but if there are some disease, for example, vascular sclerosis, then the myelin changes. Then we can see diffusion along this direction increase. So we can see that the ellipsoid model also changes. So to know the difference between 2 shapes, actually that's very easy measure of that we call standard deviation of 3 diffusion coefficients. Then we can such kinds of standard deviation is fluctuation and anisotropy. This kinds of major quantitative index. And the other one is that we know that the neural directions. The first eigenvector is actually in the same directions of the neural directions. So we can see that we can have the first eigenvector map here, and he's color coded according to his direction. For example, green left up to down, and red left to right, and blue in the through plane. So we can link the tracks by some mathematical methods. So we can have the neural track non-invasive. So that's the basic idea for us to have such kinds of neural measurement and also have the checks, calculations. And of course, the DTI is a very successful model, but we know that there are a lot of regions in the brain. For example, in the cerebrospinal fiber, in the palm regions, actually there are some critical spinal tracts from spine to cortex and also cerebrospinal fibers, from palm region to cerebellum. Then we can see actually there are a lot of brain regions that have crossing fibers. So we cannot only use one ellipsoid model to differentiate such kinds of complex structures. So there are a lot of methods that just developed after DTI to identify such kinds of crossing fibers. Then there are, the first one is DSI that proposed by my advisor, Weberdin, and also there are a lot of methods. So with these techniques, we can identify the neural tracks. For example, in TTI, you can hardly identify the regions, especially in central semioval. In the regions, there are some crossing fibers. But with the new techniques, we can identify the crossing fibers. And then we can have a very beautiful tracks map, as we show here. You can see, based on these techniques, we can identify the brain fibers, the structures, non-invasively, and even see some more detail. For example, the tracks from spinal to cerebellum, and also to identify some specific tracks we need the brain. And here we localize some specific tracks. For example, the inferior longitudinal fasciculus and so on. And here is the language pathway, and also the tracks from frontal lobe to occipital lobe, and also the Myers loop, even the very subtle Myers loop from optic nerve to occipital lobe, we can also identify it. And here is another example from papers. They just mapped the language pathway and have three segments from Broca to Wernicke and also to another regions. Then we can see the difference between three tracks, and also there are some language model that just talk about such kinds of difference, or even based on such kinds of language pathway measurement. A lot of paper they also talking about some language models to try to link with those results. And here is another example for such kinds of techniques we can use to help for neurosurgery plan. So for example, here is the tumor. You can see locates here. And here is the vascular system and also the brain structure and the neural tracks. So for the patients, when the patient have a brain tumor, they must have some treatment way. And then, for example, some patients have the brain tumor located at the motor area, but their motor function is still there. And the most important issue is that to have the patient after the neurosurgery still keep their motor function. So the idea is that to know what's the relationship between brain tumor and the neural tracts. So in this way, we can have very good neurosurgery plan. And so this technique is very powerful in such kinds of methods. And also can be used to study the lifespan changes of human brain white matter. As we can see here, the brain white matter actually developed in young population and then decline following the ages. And also such kinds of changes actually happen in different pathway, and the change, the curve is actually different. And here is also another example that we study the patients with Alzheimer and also mild cognitive impairments. We want to know what's the difference or how the brain changes in gravitic white matter. So we combine both techniques, just as I introduced to you in the very beginning about the T1 anatomy measurement and also the white matter measurement. And we want to know the progression of the structures in white matter and gray matter. Then we combine both methods, and we can see that for the red region is diffusion measurement, and yellow region is structural measurement. And we can notice that actually the change difference in different regions, and also actually the underlying axon degenerations or parameter change actually is also different in different brain regions. So the last part is the brain function. We want to know how can we measure our brain function based on these techniques. And of course, that's the idea that for us to know what is the activity of the brain function in different regions. So as we just introduced to you that actually there are a lot sources that we can detect based on different kinds of methods. But MRI is the technique that cannot measure electrical activity. Also, they cannot measure metabolic activity. But MRI can measure both functions. What's the both? What's mean both signal? That means when there is neurofiling, actually there are oxygen change, there are cerebral blood flow change, there are blood oxygen change, and also cerebral blood volume change. And most important issue is that we know that, for example, I'm talking to you, my language area is activity quite a lot, and also there are some oxygen consumption, but a lot of measurements tell us that the inflow of fresh blood or oxygen level is higher than the consumption. That is very important. There's a paper published in 2002 in Nature talking about such kinds of phenomena. So based on for MRI actually can measure all of these changes, and of course they just combine together and have such kinds of both. That's what we talk about. So both is actually blood oxygen level dependent. What's the mean? I just use this region as an example. For example, this is the neural activation region. That's the cerebral cortex in here. And we know that in this region is in general full of oxygen. That means oxygen hemoglobin. And also these regions. And the basic idea is that oxygen hemoglobin actually is a diamagnetic property. So that means diamagnetic means that when there are some magnetic field, when this material located in the magnetic field, actually they don't like magnetic line. So actually they want to just push that away. But the oxygen hemoglobin have another kinds of magnetic property is paramagnetic field, just as we show here. It will attract the magnetic lines. So actually there are different magnetic properties in this environment. So as we just introduced, that outside of the vascular system, that means the cerebral gray matter regions, we can see in this region and this region they are full of oxygen level. And for example, when here have some brain activity, then I just introduced to you that the oxygen absorption is less than the inflow. So there are a lot of fresh oxygen hemoglobin inflow. That means both regions have similar magnetic properties. So they have more homogeneous magnetic field. So the signal is higher. In compared to the regions, actually, outside of the region is oxygen hemoglobin, but inside have more the oxygen hemoglobin. Then actually there are some magnetic inhomogeneous, so the signal is less. So such kinds of idea is that there are some neural activation, then there are some glucose assumption, and also the inflow of blood flow, and also the less the oxygen hemoglobin, and also the magnetic inhomogeneous changes, then the signal increase. So that's the basic idea for MR to detect brain function. Actually, just based on the blood changes. And of course, as just Dr. Peng just introduced to you, that we want to measure brain function, we must have an evoke. We must have some input. So that's for MRI to detect such kinds of brain function, there must be a stimulus. Otherwise, when we have the brain function, you can hardly identify or hardly know what is the function mean. So in MR environment, there must have some stimulus to have such kinds of the functional detection and also to evoke the brain function. And here is an example of the function measured in elderly, and they have the verbal working memory test in young population and elder population. And you can see actually for young population, when they have verbal working memory, the area actually is less than the people in elderly. That means in this paper, they talked about that maybe for elderly, they want to have such kind of brain activity, they need more brain resource. So we can see the change a lot in compared to young population, or even when they have spatial working memory, it's almost the same. And here is another example for the patient. They have some encoding of word. So you can see in young population and old population, especially in some specific regions for elderly, need more resource. And also here is another example of functional by task in Alzheimer's patients in memory test. You can see for normal subjects actually have less. They just have some less brain resource to have the memory task. But for those patients, they need more brain activity to support such kinds of induced memory work. And then the last one is more recently that people use to detect brain function. That you use to study the functional connectivity. What does that mean? That means that actually for brain activity, there are some similar pattern in the brain. So for example, in this red region and this red region, actually they have quite similar brain patterns. So they just can claim that they have some of the functional connectivity. So people use such kinds of way to detect functional connectivity changes following ages. And also you can see for the young population, such kinds of link is stronger than elderly. And also the paper published from our lab is kinds of entropy. That means the energy changes. So also difference in different populations. So for these techniques, all can be used to detect brain structure and brain functions. So the basic idea is that it depends on what kind of question you want to answer or what kind of the source you want to detect. So here is a very short conclusion for this this lecture is that MRI actually can reveal a lot of dimensional features, for example, anatomy, neuron pathway, functional MRI, and so on. Actually, MRI and NRS I didn't introduce to you. Maybe in the coming future I have opportunity to introduce that to you. But for one of the most important issues is that nowadays actually we want to know the brain. For example, let's say an elephant, we want to know the brain, but we can only detect from different very small regions. So that's most important issue is that how to integrate all information together to have the big pictures. That's very important. So that's my lab, and thank you a lot for your attention.
Let's take this opportunity to ask some questions. Okay, no questions. The last talk will begin 4:30.